From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 30, recorded on April 19th, 2018. I'm Vincent Racaniello, and you are listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels Elde.
Hey, Vincent. Great to be here from Elde Lab Studios. I'm just catching my breath. Just was on my bicycle from a qualifying exam and then got up here just a minute ago. So thanks for waiting for me.
Hey, so when you go around campus, you ride a bike, right?
Yeah, most of the time, especially now that spring is really in full blossom here in Utah.
Do you have to lock up your bike?
Well, so when I'm here in the office, or Elde Lab Studios, I should say, we've got a nice, sort of ante room or parlor room where we've got a bike rack, and so just kind of hang them actually vertically against the wall. It almost looks like a miniature bike shop in here. Yeah, it's kind of fun. Yeah. And
then if you go somewhere, you just lock it, or
yeah. So if I'm around campus, different buildings, I've got just a cable lock that's pretty, it's not very strong, but if I'm at a meeting for an hour or whatever, it's kind of jump on and off and it's pretty safe.
You have a fancy bike?
No, not really. So I've kind of gotten, like an aging professor's bike, I would say. It's
is the bike aging or the professor?
A little of both. A little of both. So it's a city bike, kind of a hybrid. So not a mountain bike, thin tires, but it doesn't have like the kind of touring handlebars. Just good for getting around. But people take their bicycling pretty serious in my neighborhood. So tons of mountain biking trails, all this thousands of dollars, active suspension systems in order to really hit the trails and hit the wilderness, all the way to, there's a growing annual professional bike race here called the Tour of Utah, kind of, same style as Tour de France, with the stages and a bunch of professional riders. It actually goes through campus, it goes right through my neighborhood. It's kind of fun to see that action. And so I'm also just reminds me, when you see some of these professional athletes, I just feel like a different species of just sort of aging academic over here.
So we have 6 degrees and rain here in New York. It's cloudy. What do you have?
Is that centigrade you're talking
about? Sure
about?
is.
Okay.
It's not very warm. It's in the 30s here.
Okay, at least above zero there, above freezing. Yeah, we're closer to, we're right around 60 degrees Fahrenheit right now. A little partially cloudy, but the leaves are kind of coming in on all of the trees. I've got in my garden, the peas and the beans and the radishes are starting to sprout, so it really feels like we might have turned the corner. So
did you plant all this stuff already?
Yeah, I did. So
wow, that's pretty impressive.
Well, I don't know about that. It's kind of hopeful that we're maybe just sort of out of desperation, hoping that winter is over. But
we have not had warm weather that would make me want to go out and plant,
Oh, gotcha. Yep. Well, and I was talking to some family and friends who are in Minnesota, and it sounds like that massive storm blizzard came through there last week with to the tune of almost 2 feet of fresh snow. And it's sort of that heartbreaking time of the year where you're ready for winter to end, and sort of its tentacles keep kind of gripping onto you.
Yep. Oh, it's still chilly in the morning here.
Yeah.
This past Saturday, we had a science march here in New York City.
Right.
It was a big one in D.C. Did you guys have anything there in Salt Lake?
Yeah, absolute. So I would say last year, and I attended, I have to say I missed this year's edition, but last year's was really inspiring. Great turnout. We had folks like Mario Capecchi, who's in my lab, gave a speech on the steps of the Capitol. And so it did happen again. I would say a much smaller turnout. I'm guessing maybe a similar echo on your side of the coast. I've heard a little bit of rumors that it felt like it was a smaller turnout.
Yeah, so I went to the New York, last year I went to D.C. and it was great. I went to New York this year because I was invited to be the master of ceremonies.
Very cool.
With one other professor. And it happened to be a beautiful day. It was sunny and it was actually really warm, so I was in shirt sleeves. I wore a Phage t-shirt, And,
yeah, nice.
But the Washington Square Park, it was, I don't know, maybe there were 1,000 or 2,000 people, but they were very vocal. They were mostly young people, so they had lots of posters. And then when we marched, it was great because they were all chanting, but I didn't recognize a single person And I really feel I'm disappointed that none of my science friends here in New York City came out. I didn't, how cool would it be? Yeah, Mario Capecchi, he got a Nobel Prize, right?
Correct. Yep.
He's out there, and it's very cool. How many Nobel laureates we have here in New York? Why weren't any of them here, or any of the senior faculty or any of the junior faculty? Where the hell were they? Come on, this is our field. We have to get out every year. You can't let it go away. You can't let the enthusiasm go away. And everyone says, I'm busy, I have things to do. No excuse. You're not going to have a career if you can't convince people why it's good to have. So, not everyone has to turn out. But
yeah. Yep.
All right, so they asked me to help and organize it next year, so I'm going to try and get more people out next year.
Yeah, fantastic. I think and I think I guilty as charged here, incidentally, for definitely being in the mix last year, sort of slipping off the radar this year. But I think your point is a really good one, is not just sort of these every once in a while events, but to really sustain our outreach efforts on behalf of science, on behalf of the work. I 100% agree that this is part of the ongoing conversation that we have to be investing in and having.
Oh, I mean, that's why if you look at all my podcasts, all our podcasts, they sustain, right? That's totally not the case with most science podcasts. Over 95% die after 20 episodes.
Yeah, I saw that. Wasn't there a recent study?
Somebody put a paper on BioArchive that said this, basically. Of course, they didn't include our podcast because they didn't look in the right part of iTunes, which seems a little weird. But even then, we're the outliers because you have to sustain your effort. If you want to communicate, if you want to teach, you got to keep doing it. You can't quit. And the same thing with a march. The civil rights marches didn't quit after 1 year. They kept going until they got where they wanted it to get. And they keep going. And the Women's March doesn't quit after 1 year. And so we got to keep going. Don't get complacent. I know everybody's busy. You have to write papers and grants and blah, blah, blah. But take a couple of hours on a Saturday, for God's sakes. All of us have a couple of hours to donate to our field.
Yeah, that's right. And I would just, highlight that these events, at least for me, whatever they are, any outreach, whether it's the March for Science, whether it's podcasting, whether it's doing, an evening at a bar or at our Natural History Museum, all these things end up being win-win situations where I think not only is it, a great outreach opportunity to maybe get on the radar a little more with the public, but certainly in my personal experience, I always go away from these things more inspired, more energized, and sort of in positive space for pushing on our research efforts. And so even though and I totally get it, that sort of sense of, well, it's going to take some time, but carving that out ends up being, I think, both an investment for the bigger picture, but also for yourself, incidentally. Yeah.
I love marching because, everybody's chanting great chants and they have signs and people are looking at you from the sidewalk and they're pumping their fists. It's just great.
Yeah.
It's just great. You don't want to miss it. It's like
lifts all boats.
Yeah.
Yeah.
So, Nels, you have something to say about your TWiEVO on steroids?
Thank you, man. So yeah, just last quick little pitch here. It's actually, we're coming close. So I've mentioned on the last two TWiEVOs that we've got a local meeting coming together. This is a Society for Molecular Biology and Evolution satellite meeting, May 9 through 12, Molecular Evolution in the Cell. So the abstract submission is now closed. There is possibly, one or two more slots for lodging and food. It's all-inclusive, fancy Deer Valley resort, about half hour from Salt Lake. The meeting, I'm in kind of wading through the abstracts now with some of the meeting co-organizers. And I have to say, this is going to be really fun, really inspiring, amazing, groundbreaking work, kind of considering many different evolutionary questions in the context of cell biology, in the context of protein evolution, sort of a little bit of the spirit of TWiEVO, thinking about evolutionary ideas, but also testing hypothesis, sort of emerging from these ideas. And so looking forward to it. It's coming up fast.
That should be a great meeting. Yeah.
Yep.
How often do you have these?
Well, so we applied to host this and got chosen this year and got supported by SMB. They generally do several of these meetings around the country. What's really fun about them is it's a little bit of it's a kind of local regional venues, but people come from all over the world. In the case of our meeting, I think we have people from 9 or 10 different countries who will be in the crew, but it's fewer than 100 people. And so it still has a very personal, highly interactive, one-on-one conversation style meeting. And so nice. That's something that I really enjoy and think is worth investing in. Yeah. How about you, Vincent? I think there might be a promotional item on the menu coming from the East Coast.
We do. Well. We have the Department of Microbiology at the Icahn School of Medicine here in New York City is looking for faculty candidates interested in strengthening their program in virus-host interactions. Applicants should have a PhD and or MD, have postdoc experience, and be interested in creating a virus-related research group that will complement the pre-existing departmental program. They are recruiting individuals or couples at the level of assistant, associate, or full professor. More information about the school and the positions, icahn.mssm.edu. If you want to apply, you should send your CV and a brief description of your research plan to nycvirology@gmail.com, or you can contact Ben Tenhoover directly. And you know how to do that. You just go onto the department website and you can find them.
Yeah.
Easy to find anyone these days.
Agreed. Interesting fellow. I ran into him. Well, I guess the three of us ran into each other at last year's ASV meeting in Madison, Wisconsin. He gave a really interesting talk as part of the virus evolution workshop that started right before the meeting. Yeah, yeah, really fun.
And based on that talk, I invited him to TWiEVO, and he came by and talked about his dicer story. Very interesting.
Yeah, I remember. I listened to that episode and really enjoyed it. Mm-hmm.
So Nels, would you say we have a corny paper today?
Great lead-in. Yes, indeed. So as you know, Vincent, we have been sort of dancing around the comment pointed out that we've been a little bit slow to getting to plant evolution as part of the roster of work that we've considered for TWiEVO. Here we are in episode 30, and I am really excited to consider the paper today, which does have a very corny or maize-based connection. So the paper is entitled "A Kinesin 14 Motor Activates Neocentromeres to Promote Meiotic Drive in Maize." The paper was just published a few weeks ago in Cell. And this comes out of, it's a collaboration between several labs, but led by a fellow called Kelly Dawe. He has his lab in plant biology at the University of Georgia. And he also has a great Twitter handle, incidentally. It's @corncolors. Yeah, I love it. So, and this will actually connect in some real ways to the work as well. Some of the terminology here we'll spend a bit of time unpacking. So this idea of what is a meiotic drive? This turns out to be these cheating chromosomes. We have a really fascinating and I think inspiring case of genetic conflict that we'll be talking about, this idea of neocentromeres. So the old textbook ideas that we all learned as we were growing up in our biology classes about centromeres, about chromosomal segregation, Mendel's laws of inheritance. We're going to throw some of those out the window as we consider these cheating chromosomes and how the maize plants, corn, is a really historic and also vibrant system for thinking about some of these ideas and gaining real headway, experimental headway on the mechanisms under some of this genetic cheating.
Now, in terms of the authors, the PI, Kelly, he's the first author.
Yeah, that's interesting. So I think this is we talk a little bit about, some of the differences culturally between fields.
Yeah.
And so it's certainly the case, I would say, for a lot of molecular biology, cell biology, virology, microbiology, that generally, the senior author is the last author.
Yeah.
In evolution, there are, and depending on the field, there are cases where it's actually reversed, where the senior author is first.
Wow.
In this case, the corresponding author. And this sort of harkens back to the idea that sort of the person intellectually driving the work would be sort of the first one that you would see on the byline.
Interesting.
Yeah, and so I think we're getting a little taste of that.
So he did not do all the experiments.
He did not do all the experiments. He's it's actually, it looks like a pretty diverse and collaborative team. So there's and from different institutions. So for example, the last author, Evelyn Hyatt, is from Kentucky Wesleyan College in Owensboro, Kentucky. Some other labs in not only Georgia, University of Georgia, but also University of California, Davis. There's a lot of interesting plant population genetics and evolutionary biology going on there. A couple of really great labs. And then as well as the Oregon State University Department of Physics. I think that's going to get to some of the sort of molecular motor properties that we'll examine with this kinesin motor that's even in the title as well.
Missouri, too. Yeah.
So it looks like a real collaborative paper. It's certain the work itself kind of hits it at, which I really enjoyed about this work is how basically marrying different fields or crossing between different fields to really tell a fascinating story about these genetic cheaters.
So would you say, Nels, that the second author probably would be the equivalent of the first on most papers? i.e. the one who did a lot of the work and drove the project?
That really depends. So, and in fact, it would be fun to follow up with Kelly at some point. I wouldn't be surprised if he actually is responsible for some of the work.
Yeah, so he could have been. Yeah,
correct. And so some of the certainly the references of his earlier work when he trained at UC Berkeley. In a couple of different labs, including Zach Candy's lab, and in fact, who's now retired, and that's where some of the Maize project and some of the work with that Kelly Dawe had done in his postdoctoral training actually laid the groundwork for this advance that they report in this paper. And so, in that sense, he actually, I think, both a corresponding author, sort of senior author, but also has some real experimental or sort of work and genetic cross skin in the game, so to speak, as well. Yeah. Yep.
Now, Nels, can you talk just a bit about why corn or maize? First of all, is the same thing corn, maize, and what makes it a good genetic system? Because people have worked on it a long time, right?
Yeah, exactly. And so, I mean, part of it is, and this gets maybe to that Twitter handle, corn colors, and thinking about some of the evolutionary historical work, is that the cob of the corn with each kernel is sort of just a striking visual representation of the genetic processes that are playing out. And so, probably some of the most celebrated work, of course, is from Barbara McClintock and how she deduced the activity of these jumping genes, these transposons, that as part of their activity would influence the kernel color of the corn. So basically, when you pull off, and my corn anatomy is probably a little has something to be desired here, but when you pull off the leaves of the corn and you've got exposed the cob, each kernel basically is a process outcome, a successful outcome of fertilization between the pollen and the ovule of the megaspore, the female part of the meiosis that happens. And so, you have hundreds of these arrayed. And so you can very quickly sort of think about if the phenotypes are visual, like kernel color or patterning, you can start to deduce like when things might be sort of out of whack from Mendel's rules. And so if we take maybe a hypothetical example and imagine that there's a corn color phenotype that's recessively inherited. If you need both copies to sort of express that color, then you'd expect maybe that a quarter of the kernels would show that phenotype. And then when things start to go out the window in terms of the numbers, in terms of now it's not the color is stippled or interrupted. That's what was sort of led to the basis of a lot of the advances, building all the way back to Mendel's work in the crosses that he was doing in peas. A lot of geneticists, evolutionary biologists have picked up corn or maize and extended the work as we're seeing today. Yeah.
And the name is Zia maize, right?
Yeah, correct. And we'll get a little bit we won't spend a ton of time, but so this was domesticated, obviously, by humans, sort of in the rise of agriculture. The ancestral version of this, or the closest relative, is the teosinte, which is a corn-like plant, but has not been artificially selected for the kernels that have the nutrition or the other, starchy products that we now use in agribusiness that we convert into ethanol, all of these sorts of things. And so it's a striking case of artificial selection through crop breeding, selective crop breeding, which gives us modern day corn. And we sort of have this comparison point, which is the teosinte, which shares, so the corn and teosinte share a common ancestor. Not too long ago, they've both been obviously continuing to evolve, but the maize plants have undergone this crazy artificial selection and now have these, very clear phenotypes compared to what probably looks more closely like the ancestor, which is teosinte, which has sort of undergone a more quote unquote natural course of evolution without all of this selection placed on it.
I'm looking at the materials and methods, and I'm trying to find. Okay, here I finally got it. The names of the corn that they use, right? Because you'd like to know that. It's not actually mentioned in much of the paper. You got to go to the methods.
Yeah, and so maybe we should.
Is it Zia something, right? But I don't know which one it is.
Zia maize, I believe.
Is it Zia maize? Because there's some other. There are some lines like Zia mexicana, Zia parviglumis. I think they compared that when they did some sequencing.
Yeah, correct. Exactly.
Okay.
Yep. Yeah. And that's sort of, so actually the work in this project stretches back at least to the 1930s. And you can see that in some of the references. And so the observation that was made in this sort of like the early days of cytogenetics, that's another sort of really nice feature of these plants, is that the structures are large enough and that you can sort of microscopically gain access to look at the chromosomes. And what was observed, or this phenomenon was described a really long time ago, and has been considered for many years, is these sort of extra structures that appear on the chromosomes, these so-called maize knobs or knob-like structures. So the basis of this is that it turns out now that we're in sort of the genome era or post-genomic era, there are these regions along some of the chromosomes, and we'll be considering mostly this element or pair of elements on chromosome 10. So the phenotype, or the phenotype is called, or the gene area or the sort of genetic locus is called AB10, AB10, or abnormal chromosome 10. And the abnormality is that you have these knob elements. And so these are actually repetitive sequences that change the structure of the chromosome. And in more than that, can even take on this role as so-called neocentromeres. And so if we remember back from our, again, sort of high school level biology, the idea of centromeres, these are the connection points on chromosomes for the kinetochore. This is this protein complex that addresses or binds to the chromosome at the centromere. And then this allows the microtubules to then hook up as part of the spindle apparatus. And as you pull the chromosomes apart during either mitosis or meiosis, the process of forming germ cells, which will be important for the sort of cheating phenotypes that we'll be discussing. And so what has long been known is that in a lot of these corn strains in Zia maize is that they're sort of decorated, or the chromosomes are in a sense decorated with all of these knobs with different repetitive elements. Some of them start tracing their origins to probably selfish genes in a sense, so like kind of transposon-like elements. Some of them sort of related in different ways or emerging perhaps independently. But what they have in common with other transposable elements is this sort of selfish property, and that is they, in a sense, over-replicate. The knobs are access, in addition to the real centromeres. If the knobs take on this activity of being new or neocentromeres, you'll start to make microtubule connections that influence how the chromosomes are pulled together. And again, this turns out the anatomy of plants comes into play here. And so the neocentromeres, basically you end up with more connections to the microtubules, which in essence means the chromosomes are pulled together or pulled apart more quickly. The more knobs you have, the faster it goes. And it turns out, so one of the outcomes of the female germline development is that you go from, a parental genotype, 2 stages of meiosis with 4 meiotic products. And so on the male side, if it's a mammal, you end up with 4 sperm. All 4 of those sperm will get a tail. They can swim around. They can try to fertilize an egg. However, on the female side, 3 of those 4 products will actually be sacrificed. In mammals, they'll become polar bodies. They don't contribute to the germline. In plants, the 4 products called megaspores, it turns out the one that migrates the farthest is the one that goes on to become the egg. The other 3 are lost. And so immediately you can start to get a sense of how these neochromosomes, or these knobs, these maize knobs, might become cheaters. If they're being pulled, they're making more connections to the microtubules, they're pulled more quickly, they're in a more favorable position, they're more likely to become the egg that gets in the ovule, the megaspore that gets fertilized by the pollen to go on, in the case of corn, as a kernel, to then be the next generation.
So you're talking about at the chromosomal level, whichever chromosome has these cheaters, that's going to be preferentially incorporated, right?
Correct. And so these, in systems like these, are sort of broadly defined as meiotic drivers or drive systems. And so the idea is that, if under Mendelian rules, basically, and if everything is sort of going fairly, you'll do, through meiosis, you end up going from 1 product to 4, and then sort of all of the rules of the recessive, a quarter of the genes, or a quarter of the progeny will have both genes. If it's a dominant trait, half of them, all of those rules apply. Here, at the chromosomal level, you can break the rules very quickly, because basically you're ensuring that if you're one allele or one copy, a chromosome, for example, that has more knobs than another one, you'll outcompete that other chromosome.
Right. So that's why it's called the driver, right?
Indeed. So that's the drive.
So why doesn't every chromosome have a driver then?
Well, as we'll see, the farther we go, the more. And if you look at some of these classic systems, you can see that there are a lot of these cases. Yeah. There is, though, a knife's edge to cheating, right? Which is that if you are there's sort of a curse of being too successful, which is you might throw the entire system out of balance. So, for example, let's say a cheater, and there are many cases like this. Let's say the knob or the cheater or whatever the element is on a sex chromosome, and so that will determine whether you're male or female.
Get it.
And and so you can see very quickly, if you end up with, let's say, 99% males and 1% females. Yep. All of a sudden that sort of short-term gain turns into a longer-term problem that genetics will probably solve by then rebalancing the equation, so to speak.
So, Nels, what you said was there's a knife edge to cheating. Right? So I'm waiting for the knife to fall on our big cheater here in this country.
Oh man, yeah. Now we're talking.
Sorry. So I wish it were true that there was always a knife edge for cheating. In biology, there is, but in human behavior, not always. People get
not always. Although it's funny how sort of history catches up. I think in different ways. I hope so. I think we're going to find we're doing the experiment in real time in that sense, right?
So the driver is the word driver is used in different context in biology. So here it means to drive a particular chromosome into the gamete, right? We have talked on TWiEVO about drivers to get genes into a population, right? That's another name, a concept for a driver. Is that right?
Yeah, exactly right. Yep.
But when you want to get a gene into mosquitoes and release them and have them propagate through the population, you design some kind of driver, which I guess is the same concept. It keeps the gene in the population, where here it keeps the chromosome in the gamete, right?
Yeah, exactly right. And so those gene drive systems you're describing, where we're now using engineering to artificially sort of drive, for example, like the rise of an infertile population. It's sort of, and we talked about this on TWiEVO with Jim Bull a few episodes back. Right. He was doing some modeling of what might happen if you sort of allow these things to play out long term. And part of his modeling suggests that evolution will actually kind of work to somehow rebalance the equation the longer you go, and that given enough time, that even these gene drive systems that might appear foolproof will fail. You might select for self-fertilization, or you might you put a new selective pressure into the mix. But whether the short-term gain is something that is a positive outcome for something like a mosquito vector of a virus that's spreading, that could be the case. And so that's sort of also an experiment that's happening in real time as well.
Now, you want to mention the other kinds of drivers that we know about?
Yeah. So what's really cool so a lot of these meiotic drivers, and one of the features that sort of emerges from this is that you have what are called both the drivers and the responders. So these are sort of two-party systems. Another way of saying it or thinking about it in some cases that makes sense is you have a poison and an antidote. And so the poison might be the driver. In this case, the poison would basically drive the extinction of anything that's any genome or chromosome or element that's vulnerable to it. However, if it's linked to a responder or an antidote in this case, then the thing encoding it might enjoy resistance to it. And then you can imagine a very rapid case of cheating where if every other sort of genotype is vulnerable, if you don't have that responder or that antidote, then that will drive to fixation really rapidly. And so some of the famous examples of these that have been most studied, and again, we kind of echoed on a little bit on an early episode of TWiEVO. So my colleague Nitin Fudness, who's in the biology department here, he thinks about things like segregation distortion in Drosophila. This can lead actually in cases to a skew in sex ratios. So you end up with more female flies or more male flies, or the males might be infertile or the females might be infertile, depending on the system. And so people have, in many cases spent a lot of time and investment, more than 50 years working on some of these, and with the new tools revealing some of the genetic mechanisms at play. There's also this in mice, this so-called T haplotype system. And it is a case where, so now the sperm coming out of these mice with the T haplotype actually have a modification to the microtubules that changes the ability of the sperm's motility. So you can imagine if you sort of stack the deck to be a super swimmer or if you're not a good swimmer, then again, you sort of have the ability to cheat. What's really cool about today's system, this abnormal chromosome 10, these Maize knobs, is that it's sort of a subset of these meiotic drivers. It's something that's been called centromere drive.
So there could be different mechanisms of drive, right? That's what you're getting at.
Exactly right. And so for those first cases that we've just been sort of discussing a little bit, those are happening sort of post-meiotically. So you've already completed the meiotic process. You either have all of your sperm in place, all of the eggs are in place, and then you're just sort of saying, okay, let's let these things get together and fertilization to occur. These cases of centromere drive, which are noted now in several systems, including mammals, in this case, the process of meiosis itself, so the way that centromeres are defined or recognized by things like the motors or the kinetochore proteins, the complexes that hook up the chromosomes to the microtubules to actually undergo the separation. The cheating or the sort of the process of that happening in an equal manner is now is actually being altered in real time, and that's what's going on with these Maize knobs. So you're changing basically how the microtubules and the motors are connecting to chromosomes in specific ways, and so that's a case of the so-called centromere drive. They
say these are pretty rare, these meiotic drivers, right?
Yeah, that's right. And so, although I would say that we are starting, like folks are starting to sort of observe the phenomenon in more in interesting cases. So in addition to my colleague Nitin, he and I were both postdocs with Harmeet Malik, a guy whose name comes up from time to time, both on TWiV and TWiEVO. He actually started his lab pointing out that if you look at the evolution of centromere proteins, so these are the proteins that sort of help define or organize the chromosomes into the centromeres, that he and when he was a postdoc with Steve Henikoff, they made these sort of really interesting observations of all of this rapid evolution in the centromere genes encoded, for example, by primates, encoded by humans, and in some other systems, and then inferred from that something like a cheater system or an arms race scenario.
Right.
And so in that case, the idea is the centromere's identity is changing. And so more and more examples of this, some other recent work from Michael Lampson's lab in mice that might be getting at some of the mechanisms there. He's at Penn. And then very much a similar kind of flavor or idea or concept, but with a very different biology in some ways with the neochromosomes that you see in maize that we'll be talking about today. Yeah.
So where does this paper pick up?
Yeah, good question. So the paper itself picks up, so this abnormal 10 has been well studied, including by Kelly Dawe's lab, and they want to tackle the question, which is basically they know what was known was the sort of DNA basis of the repeat elements that form the knob. So there's two sets of repeat families, the so-called 180 base pair repeat, which they'll focus on, and another set of repeats, the TR1 family. And we'll get back to that in a moment. So that was known, that what the DNA elements were. But what wasn't known was basically how this knob, this structure in the chromosome, becomes active or takes on the action of a neocentromere or a new centromere. And in order for that to happen, there has to be some physical connection, a protein or some element that then hooks up to the knob and then and adapt as an adapter or a motor that then pulls on that region of the chromosome to move it more quickly out towards relative to other chromosomes that just have regular old-fashioned centromeres, and then brings it forward.
So that's the connection between the chromosome and the spindle, right?
Exact. And so that was the question they wanted to ask, is basically, what is that connection? What is the function, the gene encoding a protein or a related function that executes this in this system? And so what they did to attack that question, it was pretty clever. So one of the things about these derived systems, and especially the two-part or at least two-part systems, is that the two entities are linked, right? So if you encode a poison and an antidote, you probably want those two things, if it's going to be successful, those things are probably going to be in close physical proximity, either right next door to each other on a chromosome or in the neighborhood. And the reason for this is because of recombination. So as soon as you divide or recombine the antidote away from the poison, then the poison is sort of unleashed, and so there's a definite sort of evolutionary imperative to keep these things together. Or in essence, the things that survive are the ones that don't get broken up. And so using that reasoning, the authors of this paper basically sort of had the idea that whatever is encoding the ability to make these knobs into neochromosomes must be nearby in the neighborhood. And in fact, in a lot of these systems, people are observing, now that we're, again, in sort of this genomic era and sort of looking at the larger architecture, that they're around these cheating systems, these driver systems, are a lot of inversions. And relative to sort of like the wild type chromosome, so to speak. And the reason that these things are probably enriched, or the emerging idea, is that when you have a bunch of inversions, it actually makes recombination just mechanically more difficult to execute. And so you, in essence, lock these regions of the genome together. Yeah. So anyway, using that kind of intuition, the author said, well, we have all these strains, and one in particular, abnormal 10 strain that had a deficiency in, so basically a pretty major, I think a 1 megabase deletion of DNA that was right next door to the knob element in that strain on chromosome 10.
And that did not have a drive, right?
Indeed. So it still has knobs, but it's not a neocentromere. It doesn't drive. It just kind of hangs out. And so the inference is that, well, maybe something in that 1 megabase is, encoding the function that is actually the sort of player that they're looking for in this two-part system. So this is where kind of the TWiEVO hot space that we're looking for is using experimental or technological genomic tools to attack that idea. And so what they did is a pretty simple comparative RNA sequencing experiment where they just take tissues from either the abnormal 10 strain that has the cheating activity or that deficiency strain. They isolate total RNA and then compare what is missing in the deficiency strain relative to the abnormal 10 strain, the neocentromere. And so what they found was that they pulled out a number of differences and then sort of tried to confirm that those differences were real, that basically there were messages missing in the deficiency that were found in the neocentromeric strain. And I think they found about a dozen that they confirmed by PCR. So basically that they could get the RNA messages, the transcripts from the abnormal 10, but were missing in the deficiency. Of those 10, one of them was a gene. The others were sort of non-coding regions. And the gene that they found was sort of already put them onto the idea that it was a really strong candidate for the activity that they were inferring. And that's because it was this kinesin 14-like motor protein. And we can talk about why, and they'll get into some experiments for why this might be the responder in a sense, or the second part of the system, a protein or a motor that could hook up potentially directly to these elements and then cause the chromosomes to be pulled into the megaspore and to outcompete their knobless or relatives
Yeah. Nice. Yeah. Nice.
Yeah, I mean, I'm kind of curious what the other 9 regions are. Are they real? Was it background?
Sure.
Or
because they said they were not coding for anything, right?
Yeah, exactly.
Maybe long non-coding RNAs or small RNAs could be interesting, right?
Yeah, yep. So they're kind of, now that they have this, so the just by looking at the sequence, there's enough known about these kinesin motor proteins, and so these are proteins that they will attach to cargo on one side of the protein. On the other side, they can hook up to microtubules, and then they have a direction. They're either plus, so microtubules have a polarity. They either have a plus end or a minus end. And this family of proteins, which have been described in a lot of experimental details or biophysical detail, is known to be a minus end directed motor. And that immediately fits just with the biology. So it turns out that, when you're forming a spindle and pulling chromosomes apart, whether it's mitosis or meiosis, the kinesins, the motors that are generally known to encode this activity are in fact minus end movers. And so that's just the polarity of how the spindle is arranged. Another detail that immediately sort of fell out from identifying their candidate gene was that the kinesin sequence was not alone. So it was actually in a number of copies in that deficiency region nearby the knob elements, the 180 base pair elements. And so there could be as many as 8 or more copies in some state of repair or disrepair, depending on the strain that you look at. Mm-hmm. And so they ended up naming that locus Kinder, Kindr, and that's short for kinesin driver.
Of course, it also means children in German, right?
Yeah, right. I'm curious if they say Kinder or Kindr or how they pronounce it. But so even more than that, actually, they were able to again lean on the tools that are available, given that so many people have, so many labs have studied these maize strains, and there are some that are known to be suppressors. So they still have the knob. And all of the elements are in place, and this sort of cytogenetics, you can look at the chromosomes and you can make out these structures under the microscope. However, there are these suppressor strains where you look at them just in terms of the morphology of the chromosomes, and you would say everything's in place. They should probably be doing this sort of neocentromeric cheating activity, and yet they don't. And so they took those strains and immediately asked, what's going on with that kinesin, the Kinder, that locus, and the copies of the kinesin gene and the repeated copies of it. And what they found is that the expression in those suppressed strains of it was much lower. Mm-hmm. And they could trace that to the idea, or correlate that to the idea that those regions are heavily methylated. Mm. And so that, in plant sort of epigenetics is a sign that that chromosomal region is probably going to be under low expression. And they were able to confirm that in their experiments. And then the really nice touch, they went on and actually said, okay, well, let's see what happens if we experimentally silence that region. So they used RNA interference, RNAi, to target that region. And what they discovered was that they were able to knock down the expression of those kinesin genes. They turned out to be methylated, so a similar mechanism as the suppressors that they had already isolated or had just characterized. And lo and behold, that RNAi-treated, now methylated and turned down expression also does not act as a cheater as much as the wild-type strains. And so now at that point, they announce fairly confidently that they've figured out what is the connection between the maize knob and the machinery, the responder, the two-component system, in order to have the centromere drive system in maize.
So this RNAi was directed against Kinder, right? Mm-hmm. You get reduction of Kinder expression, and then you don't have the meiotic drive anymore. And
correct, or that's right.
And
it's greatly reduced.
Reduced, and they call this an epimutant, right?
Yeah. And so the idea there is that because it's methylated, and that kind of state can be inherited. Yep. In fact, it could be transmitted, from sort of mother to offspring. Yeah. And might sort of continue to go. Because
epigenetic means like modification of chromatin or DNA even, right? Not changing a gene, but actually its expression.
Yeah, correct. And so, we won't open that can of worms just because there's many sort of competing definitions of epigenetics in some ways. But yes, that's in this case, in that sense, very much so. I mean, one of the questions that's raised for me, which I think is, and I'm curious whether they're sort of pursuing this, is that, when you look at the expression data itself, either in their RNA knockdown strains or those suppressor strains that they naturally characterize, and they look at, for example, the leaf tissue, they look at the anther, so that's the male part. What you can see is that the relative expression that they measure is lower, but it's not zero. And so this, kind of got my curiosity going, or thinking, is it that there's sort of a threshold here where it's not kind of like flipping a switch on or off with the motor, but you need a certain amount of the motor to see the activity? Or I'm just kind of wondering if there's a threshold. It'd be fun to see where the story goes, whether there's more to it.
Yeah, I mean, the motor is supposedly bridging the knob with the spindle fibers, right? So maybe you need a certain number. Would be interesting to do some interaction studies. I guess they're probably working on that, right?
Yeah, that's correct. And they also point out, and it's not a small detail, which is that they currently don't know whether, they'll show some co-localization data in a moment, but they currently don't know if the interaction between the DNA elements, the knobs in the kinesin motor, whether that's a direct connection, meaning DNA to kinesin protein motor, or if it's an indirect action. There could be an adapter or a bridging protein or set of proteins between the knob element and the motor. And they haven't formally ruled that out. You could imagine, and maybe they're doing this or have already done this, pull-down experiments.
Yeah, sure.
Right, with to see whether they get more proteins. You could do that like a quick pull-down. I say it now as a PI, quick, any experimental setup as quick. But a pull down and then follow that up with mass spec to identify any potentially interacting proteins. Or is it really nothing else there? It's just a protein DNA interaction that then gets the neocentromeres onto the spool. Yeah.
Well, they did do some immunofluorescence microscopy, right? And they could show that the kinder protein is at neocentromeres, right?
Yeah, exactly right. So this is in figure, let's see here. Actually, is it figure 2?
It's
figure 5. Okay, yeah, we're farther along here. Figure 5. So in panel E, you can see, and this is actually, they took advantage in several places in the paper. I think this is a really nice sort of internal or natural control in a sense. So there's the 180 base pair element. That turns out to be the one that's interacting either directly or indirectly with kinder, the kinesin motors. But then there's sort of associated with these knobs are these TR1 repeat elements, which themselves could be the basis potentially of a neocentromere with perhaps a different interaction. And the reason this is a nice control is that so they show, for example, their the kinder localization, it overlaps to a large degree with 180 base pair repeats, but doesn't overlap with a TR. So there's some specificity here. Yeah. And that also hints at how you can get all these other sort of drive systems kind of going simultaneously. So it's like kind of multiple cheaters with different responders or different component systems that can arise independently. And then you get into even more complicated scenarios where, if you're team a couple of these up against another one and who's stronger than who, that could sort of have an outcome for the inheritance here outside of Mendel's rules.
The other experiment I really liked is this microtubule gliding experiment.
Yeah, exactly. Yeah. Yep.
Where they can actually see in vitro, they can assemble microtubules and look at the protein moving on them. Because the kinder is a kinesin, it's a motor, right? You can label them up and you can measure the speed of movement on microtubules.
Yeah, exactly. And this is super cool because first of all, it confirms that these are active motor proteins, so it sort of helps connect the mechanism. But then there's also like I think they did a nice job here of taking advantage of sort of an evolutionary comparison. We'll talk about this a bit more. But so, you can immediately, once you have the identity of your candidate gene, compare it to others. So it turns out the kinesin sort of paralogous family of proteins is large. And then so you can ask, what's this one most closely related to? And so the closest described kinesin before they discovered Kinder was one called kinesin 11 that's known to be in plants, also minus end directed. And so then they can compare in vitro the velocity or how strong or how fast the motors are. Mm-hmm. And what they found, this is again in figure 5 panel B, what they found is that the Kinder motor looks like it's actually quite strong. It has almost twice the velocity of the. And then, so this kind of gets you thinking, right, about the mechanism again of cheating. Yeah. Knowing that the faster you go, the more likely you are. And so now you can imagine that sort of natural selection has acted to take the ancestral or maybe, common ancestor of those two kinesin motors, and for Kinder has selected for ones that can really cook along the microtubule.
Yeah, it's great. It's really cool.
I like that. Yep.
I guess you could make an altered Kinder that doesn't go as fast and test that, right?
Yeah, exactly. So, I mean, it would be, probably not a trivial experiment to do get your recombinant strains together, but I think you would have that strong prediction, a really cool hypothesis that would, as you sort of compete, if you could figure out how to compete, either separately in different strains, that you might see that the cheating activity would be higher if you had, if it was speedier, had a higher velocity. I think that follows nicely from the data so far. Yep. Yeah, and then sort of the final turn for the paper is that kind of building on what you can do with a comparative approach. And you mentioned sort of in the methods, right? They considered or compared the Kinder sequences. First wanted to ask, when did this thing arise? Yep. And so you can trace back by looking at other species and asking, is it in this system? Is it in this species? Is it in the strain? And basically, if you correlate that with molecular clock estimates, they propose that this thing is about 12 million years old, that Kinder came online or duplicated from another kinesin owing to the fact that it's missing in species that are thought to have diverged more than 12 million years ago relative to maize. Mm-hmm. And so that's where those other species in the genus Zea were useful to then compare and ask, are there differences among the in the Kinder loci and even in the coding sequences? And so I just mentioned briefly when we were discussing centromere drive that one of the kind of landmarks that Harmeet and Steve Henikoff noted or discovered, put forward, was this idea of rapid evolution or repeated positive selection of centromere proteins, which sort of is a surprise given that they have a pretty important housekeeping function, which is to properly segregate chromosomes. And you would think sort of maybe a priori that these were the things that would be conserved because it's kind of a core function of chromosomal dynamics. And yet these are some of the fastest evolving. And so the authors here look at Kinder and ask, is it also undergoing rapid evolution? And at least from the comparisons they made at that sort of amino acid substitution level, maybe a few residues, but nothing to kind of write home about. Mm-hmm. And so at the same time, they do note that this is sort of duplicated in tandem arrays. Some of the copies look like they're intact and functional. And in fact, they sort of, show really nice evidence for that in those microtubule in vitro assays that we were just discussing. However, there is some volatility here. So it could be that there's more to the mechanistic story that, depending on how many copies you have, maybe that threshold effect. These are all, I think, open questions for how the system is working and how it might be regulated. And there might be some clues just by taking that comparative approach. Mm-hmm. So then, Vincent, I think the fun sort of final part of this is how they really
cool.
They kind of they then consider for a minute what is the evolutionary impact of these cheating? Like, can you start to account for how sort of prevalent they are? If you again kind of comparing species and make an estimate of how much cheating has sort of unfolded here based on how prevalent these things are, and so. They point out someone had, noted earlier that the 100 base pair repeats. And so remember, when you form these neocentromeres, the genetic elements, whether it's genes, other just genomic regions that are closest by, and in some cases, in essence, locked in because of these inversions, those are the things that are going to kind of co-segregate with the cheaters again and again, and will sort of sweep up in frequency in populations. And so someone had published that about 8% of the maize genome consists of the 180 base pair repeats in sort of the neighboring sequences. When they took a closer look at their strains, they're proposing that might be as high as 20% of the genome.
Amazing.
Yeah, and so this is a good cheater, and they estimate that to be 500 megabases of sequence that has sort of outcompeted other sequence. That's bigger than many genomes altogether. So Maize is 2 gigabases. To put that in perspective, the human genome, 3 gigabases or 3 million base pairs, and this is a quarter of the whole Maize genome. Just sort of showing up by just the selfish activity. And so, there have been these ongoing debates in the field. So like when the ENCODE project came out, how much of the genome is functional, how the whole concept of junk DNA, which is sort of a lightning rod for what the definition of junk is versus garbage versus so on and so forth.
Yeah.
And yet I think what this, again highlights and demonstrates is that just with a cheating mechanism, that massive parts of the genome can sort of be overrepresented or can quickly sort of take over genomes or expand genomic real estate. Just with, it is functional, but it's just a selfish function of outcompeting in other chromosomes without the elements in that case. Yep.
The inevitability of genetic parasites.
There you go.
And Eugene Koonin said this to us. He said, I think when cheating is possible, it will happen. Something like that.
Yeah, and I think what we're just glimpsing at is how so it will happen, but also how sort of far-reaching that impact can be.
Yeah.
So here you have to start, redefining or accounting for how this violates the rules of Mendelian inheritance, how this has a massive impact on chromosomal structure, and there's even some work from some population geneticists that this is Graham Koop and Yaniv Brandwein, who's now at Minnesota, suggesting that they think these cheating elements in some of these plants might influence how recombination is occurring. Again, because you have sort of the dispersion of these inversions, and you sort of set up, in any of these cheater cases, there's sort of a second side of that coin, which is how do you sort of survive or suppress the cheating or rebalance the equation. And so a lot of recombination in plants might be in sort of where the events are happening and the frequencies at which they are, might be a direct outcome of these kind of parasitic elements.
It's amazing.
Really interesting. And I think it just kind of highlights again how our textbook view of the rules of biology are sort of constantly challenged or violated.
As they should be.
Yeah, exactly. And sort of revised.
Yeah.
For me, I think the work, I don't know, it's hard to just kind of put my thumb on it, but there's something kind of almost poetic to me in how you have these kind of historically studied systems, the great phenotypes, the kernel colors of corn. They actually have a picture in one of the figures. This is in figure 4.
Yeah.
Where you have the abnormal 10 has a lot of the dark kernels, and then that RNAi strain they made, you can now see more yellow kernels. There's not as much cheating happening in that strain. And so you have that sort of very visually striking phenotype all the way to sort of, how the genetic puzzles that have kind of grabbed the imagination of population biologists for a couple of generations. And then you mix that with the emerging tools, the genomics, the RNA sequencing comparisons, the ability to really understand chromosomes just both at the kind of massive scale all the way down to single nucleotide resolution, and how that's allowing us to make real progress on understanding these systems sort of at every level of biological organization.
I really like this paper because it explains, how the cheating works, how these knob reach, how these neocentromeres work, but it also reveals all the work there is to be done still, which is great.
Agree.
For me, had helped me to catch up on Maize because I haven't looked at a Maize paper in a long time. Have you?
Yeah, no, it's been a while. So, kind of when it rains, it pours. So we were considering several plant evolution papers that have come online recently. One on auxin evolution, sort of the plant hormone that's sort of classically studied. There's some really cool recent work on that. This one, some cheaters, and several other papers that are just emerging. But sometimes it falls off the radar a little bit, but always reminded that the biology in plants, in some cases like immune defense mechanisms, evolution there, cases like this, where you have all of the tools, the ability to do crosses and sort of control the system, like there's just interesting breakthrough after breakthrough. And in systems that I think, we're do a lot of navel gazing at our own species and invest a lot in our own health and sort of fighting diseases, genetic or infectious, that our species is dealing with. But to remember that, things like the things we eat every day and how we sort of use biology and technology as a way forward with increasing global challenges to food supply, this is just as important. And so it kind of gets short shrift sometimes compared to the both I think the scientific, but also the value and sort of necessity for society going forward.
You bet.
Yeah.
Let's see. You want to read a couple of emails here?
Yeah, let's hit the mailbag. Do you want to do the first one?
Sure. This is from Trudy. I found your discussion about the neutral theory of evolution on TWiEVO 25 very interesting. How does this theory tie into hard factual data supporting selection, such as those generated by Peter and Rosemary Grant in the Galapagos Islands over the better part of the past half-century? The Grants have clearly shown that the finch's beak sizes change fairly rapidly in response to changes in the food supply. Nels, congratulations on your tenure. Thanks to both of you for your continued efforts with this wonderful podcast. Kind regards, Trudy.
Yeah, nice. So thanks, Trudy. And also thanks for bringing up the neutral theory in our sort of brief discussion. This kind of also highlights to me that we should, we're a little low, I would say, on TWiEVO episodes in the category of population genetics. And there's a lot that we could add to the conversation of active work going on there. And so we'll kind of keep our eyes out for some recent work that's kind of constantly emerging that might be fun to have a guest or consider going forward. I would say just kind of quickly that, so actually it turns out that these things, so the idea of the natural selection, this is another striking phenotype, the beaks of the finches, right, on the Galapagos. So the idea being that as the ancestors of those species showed up on the island and they sort of were competing for different food sources or finding different niches, that you end up with these differences in beak morphology that are the difference between being able to crack open a different nut or reach into a space to get something that's a really striking case of natural selection. Mm-hmm. So that actually doesn't collide or conflict with neutral theory. So neutral theory or near neutral theory, sort of a advance or a modification of it. It's the name can be a little, especially for a non-population geneticist, and I put myself in that category for the most part. The name can be a little sort of intuitive or counterintuitive in a sense, where the idea of everything being neutral, that's not actually what the theory says. So what it does say is thinking about, and hopefully we'll get this at least partially correct, is that if you just sort of take mutations in general and sort of look at the overall population level, that most mutations will not be sort of lead to a case where natural selection will act in an adaptive way really quickly, which would be the case underlying the finch beak. And so the neutral theory has plenty of space for natural selection to act. Right. But it's sort of thinking about, kind of the generic, or not quite average, but generic mutation, which would most likely kind of given things like the degeneracy of the genetic code, the robustness of protein folding, that most mutations will probably not have a massive impact on a protein function, or part of the idea is this in the near neutral theory is that they may be slightly deleterious, that you sort of go away from kind of an adaptive peak, and that a mutation that does change sort of protein function slightly will maybe throw things a little bit out of balance. Yeah. Yeah, and so then it starts to get into population sizes and kind of goes a whole different population genetics way. But the good news is there's room, so that there's still active work going on here, and there's plenty of room for adaptive natural selection outcomes as well. It's just some of the bigger questions. Yep.
I'll take next one.
Sounds good. Josh writes, "Hey, love the show. Interesting article on viruses and how we form memories. Great animation. Keep it up." So there's a link here, and I'm guessing this is to my colleague, Jason Shepard, who's at the University of Utah. He recently had a paper on this gene ARC, which derives from endogenous retrovirus envelope.
A retrotransposon.
Yeah, this has gotten a lot of heat in the press. That definition is important. And the gene appears to basically take on a role, or the protein may be involved in forming these structures, exosomal structures that have a role in neuronal synaptic transmission or neuronal communication that influences memory. Yeah. And so the memory side of it, that's been known for a while for the protein. It comes from things like mouse knockout experiments where on memory tests, these mutant mice suffer some consequences. But then Jason's work that Josh is lifting up here connects back the evolutionary origins, which was a collaboration with another colleague of mine who's now at Cornell, a fellow called Cedric Fichat. He and Jason teamed up and sort of looked at the evolution of ARC, finding that retrotransposon connection to the envelope gene that then was they proposed to be co-opted for this now memory function. Really interesting. And you discussed on TWiEVO.
477 Raiders of the Lost Ark.
Yeah, how was I haven't had a chance to listen to that one yet. How did the discussion go? Yeah, cool.
It was, we were really impressed. It's an amazing idea. I mean, I think it needs a little more work, but it's amazing that this GAG protein, which is from a retrotransposon, not a endogenous retrovirus. So the retrotransposons are precursors of endogenous retrovirus. So they don't have envelope proteins, so they're not viral, but they can make particles. And as you said, they may be involved in synaptic transmission. It's pretty amazing.
Yeah, really got a lot of attention too.
Stuff, yeah.
Yep.
Last one's from Larry. Muller's ratchet versus Amazon Molly. Amazon fish challenges mutation idea. This is an article in BBC News. I'm still catching up on TWiEVO, so apologies if you've covered asexual reproduction in animals before, maybe the escaped crayfish. Also, I haven't read the original paper, and I'm not really qualified to judge it anyway. I'm just a biology fanboy. I'm an IRL software engineer, but the BBC article by Jonathan Ball sure makes it sound like this study of the molly raises interesting questions. Background: I love the podcast, even though many of the details are lost on me. Nick Lane's Life Ascending kindled my interest in evolutionary biology, which led me to TWiM and then TWiEVO. Thanks, keep up the great work. Larry's in San Diego, and then he writes, P.S. Now reading Lane's The Vital Question, a deeper dive into the mechanisms, and I just finished TWiEVO number 20, wherein you sound about as enthusiastic as I am about Nick Lane's books, which was vicariously satisfying. 2 additional comments. 1, you are really doing an excellent job communicating. The mix of casual, serious, and amusing is perfect. 2, I think it was in this episode you mentioned advanced high school students listening but not getting all the terms. It might be helpful if the microbe TV sections for each episode listed a few terms or acronyms, initialisms that aren't easy to spell from their sound. Not full definitions, just the terms. Anyway, love the show and thanks again. So this article is very cool. It's something we just talked about yesterday. I gave the evolution lecture in my virology course. And I talk about Muller's ratchet, which was an idea, put forth by Manfred. No, not Manfred. What was Ratchet's name? Anyway, I can't remember his name. It was Professor Ratchet. Muller, Professor Muller, sorry. Ratchet was Nurse Ratchet.
Herman Mueller, maybe?
Herman Mueller.
Yeah.
Basically, the reason sex arose, he thinks, is because asexual populations, they just keep accumulating mutations because they're basically cloning, and they eventually burn out. They die. But then they found this molly that they estimate to be 100,000 years old, and it apparently does asexual reproduction, which is very interesting. Although it needs sperm to touch the egg, to get it going, the sperm doesn't get incorporated into the fish egg, and this fish has been around. And so what's going on? And, they say, one of the ideas that they say in this article is that, the deleterious mutations just go away because the fish die, right? And they never get propagated. But there are also other ways that the genome can change, as we've learned, right?
Yeah, absolutely. Yep.
It doesn't have to be through sexual reproduction.
Yeah, no, exactly. And I think it's really fascinating. It also raises the question, again, that we kind of discussed a little bit, the long-term versus short-term, sort of an evolutionary standpoint. So this one sort of is surprising that it's been around, but come back in 1 million years and will it be around? Or do those populations become more vulnerable? And this gets at The idea that having recombination or sexual reproduction is the idea of the Red Queen, that this gives host populations that might be infected by parasites, viruses, otherwise, that to kind of stay even in terms of lower mutation rates, longer generation times, et cetera, that recombination sort of levels the playing field. And so the idea that emerges from this, any asexual population, I love that crayfish story that came out a few months ago in pet stores. How long will these are these evolutionary dead ends, these populations that sort of go into asexual, will they become more fragile or vulnerable to future challenges? And it will all the environment will matter. Does a parasite come along or whatever? Yep. Really interesting. So, and also thanks, Larry, for both the compliments and the suggestions. A really good point on the acronyms, the initials. That also just reminds me of something I wanted to mention with, including the papers that we sometimes, if they're not sort of open access, this one isn't, it's in the Cell Press family. But there is, so if you really do want to follow up and kind of get a handle on the alphabet soup that we're sometimes talking about and really see the work for yourself, a very simple suggestion is to just write a quick email to the author and just request the work. And I think vast majority of the time, myself as an author, when I get these requests, I will within 24 hours send a PDF copy, and am very sort of encouraged that people care enough to reach out and ask or request to look at the work. And so that's another way of getting access to the details as well. Yeah.
All right, Nels, I have about 10 minutes. I have to have an office hour, so let's do some quick picks.
You and me both. I've got a meeting at 2 as well, so we're at 4 o'clock. So my pick this week, science pick of the week, is but for me, it was an interesting sighting of nature on the internet. Of course, just sort of the Twitter channels or Pinterest in this case, I guess. Mm-hmm. And this is a fungus, a mushroom called the craypot stinkhorn. It's from Australia. I included there's a link here, but I also included a photo in our show notes, and I just thought you might enjoy the morphology if that structure reminds you of anything.
Love it. Looks like a virus to me.
Yeah, exactly.
It's a buckyball, I guess, right? But
yep.
Clearly viral. Yeah, it's great.
Yeah, so you get these.
I love it.
These patterns that emerge at different sort of levels of biological organization, whether it's a mushroom taking on the appearance of a virus particle, or maybe a clathrin-coated pit, or a exosome or something.
I guess it could be a clathrin-coated pit too, right?
That's what it reminded me of as sort of cell biology background.
Pretty cool.
But anyway, I thought that was kind of a fun sighting of cool structure, biological structure that you can see at the molecular and also at the sort of macro level with these mushroom species. How about you, Vincent? What's your pick of the week?
So I have an article in Nature which is entitled "Doubts Raised Over Australia's Plans to Release Herpes Virus to Wipe Out Carp." So Australia has a history of trying to wipe out species with viruses. They first tried it years ago. They had imported some rabbits from Europe, and the rabbits overran the country, so they introduced a pox virus, a myxoma virus. And it didn't work because after the first year, the virus and the rabbits evolved, and the rabbits are still around.
Oh, yeah.
And I think they tried it for some other species as well. And now they want to do it for carp, which apparently escaped from fish farms and got into the local rivers when they flooded. And now they're overgrowing the rivers and they want to kill them with a herpes virus. And we covered this on TWiEVO last year, I think, where they figure if they infect these fish, they'll all die. Then they'll have these millions of pounds of fish in the rivers, which have to get rid of because they can't let it rot. So it's a big problem. But now this article says it probably won't work because the water's too warm and the virus doesn't replicate well. So it sounds like they might not do it, which I think would be a good idea not to do it because whenever you try and do this, the virus and or the host both can evolve so that it doesn't work.
I agree. That's definitely the case. Evolution happens, right? So that was
Evolution happens. That's right.
That was the case with the myxoma in the rabbits, and you can imagine a similar outcome with the carp. Yeah.
All right, cool. That is TWiEVO. Hey, number 30. We got to a nice number.
Yeah, pretty great. Having fun.
You can find it at Apple Podcasts, microbe.tv/TWiEVO. Do subscribe on your favorite podcast player. And if you'd like to support us financially, go to microbe.tv/contribute. Send your emails to TWiEVO at microbe.tv. Nels Elde is at cellvolution.org and on Twitter, Earlybird. Thanks, Nels.
Hey, thank you, Vincent. Fun to be in touch as usual.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to Trampled by Turtles for the music on TWiEVO. You can find them at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious.